Hi, my name is Morgan, and today I am going to be talking about feeding the growing population, use of genomic tools to improve food security. So, here's some general facts about the food system and food security. It is important to keep these in mind so that we can understand how great of an issue food insecurity is and how we can improve food security. So, today it's estimated that 821 million people are undernourished and 151 million children under the age of 5 are stunted. This means they have impaired growth and development. I have also included the 4 pillars of food security, and the one I want to emphasize is stability. How can we stabilize the supply of food, making sure that everyone has enough food all the time? How can we improve food security? So, to understand how to fix the problem of food insecurity, we have to understand why it exists. So, why exactly does food insecurity exist? There are so many reasons, but one part of it is because there is simply not enough food. Then we might ask ourselves, well, why isn't there enough food? It's because crops are being killed by environmental factors like drought, as well as pests and diseases. Currently, we have things like chemicals to prevent pests from attacking plants and host resistance to prevent disease from killing them, but chemicals pose environmental threats and pathogens are constantly evolving, so neither of these offer a long-term solution. While we might not be able to make sure that all 821 million people are fully nourished, maybe we can make sure that 500 million people are by increasing the amount of crops produced. So, one long-term solution is genome editing technology. Dr. Kumar defined genome editing technology as a collection of advanced molecular biology techniques that facilitate precise, efficient, and targeted modifications at genomic loci. We can modify the genome using insertion, deletion, and point mutations. However, we have to understand some basics of genomics before we can understand how this can change the genome. So, what exactly is genomics? So, a genome is the set of instructions needed to create an organism. It's basically the cookbook for making the desired product, whether that desired product is a plant, a human, an animal, or anything else. Genomes are stored in the cell as DNA, and DNA stores the information needed to create the organism using a special code. This special code is the order of nucleotides. So on the left I have a certain order of base pairs and on the right a different order of base pairs. Because the order of the nucleotides are different, these sections of DNA are going to do different things. Just as an example to clarify, the order of base pairs on the left produces a protein that allows a plant to do well in drought conditions, but the order of base pairs on the right produces a different protein that does not help the plant in drought conditions. Genomics is all about how the genome interacts with its environment. So today there is still much unknown about how we can edit the genome, but here are just a few ways that currently exist to edit the genome. These are all super in-depth processes, but they all have the same basic job of altering the order of the arrangement of nucleotides in the DNA. To the left I have shown a picture of zinc finger nuclease. On the right I have a picture of transcription activator-like effector nuclease, meganucleases, and clustered regularly interspaced palindromic repeats are also two other ways to edit the genome. One of the modern genomic editing techniques of special importance is clustered regularly interspaced palindromic repeats, also known as CRISPR. It uses a protein called Cas9 to change the original arrangement of nucleotides. The protein can change the arrangement in many ways, including indels, insertion, transcriptional repression, silencing, base editing, and epigenetic editing. These each alter the arrangement in different ways, but they all have the same goal of changing the order of nucleotides in DNA. Because if we can change the order of nucleotides, thus changing the structure, we can change the function. So, changing the genome might offer a long-term solution to issues that hurt crops like drought, pests, and disease. One example of a crop that is being negatively impacted by drought is soybean. If we can improve drought resistance, we can improve soybean production. Soybean is one of the most important leguminous crops. It is well known for its protein and oil contents. Over 70% of the soybeans produced in the United States are used for animal feed. So while soybean isn't directly being consumed by people, a large portion of animals' diets is soybean. Without soybean, we couldn't feed livestock, and without livestock, people would starve. Drought stress threatens soybean growth and productivity by impacting growth at morphological, biochemical, and molecular levels. Drought stress can reduce crop yield up to 40%. It is one of the most crucial environmental factors that reduces crop yield. We need to develop soybean crops with drought-tolerant traits. Now I want to talk about how we can use genome editing technology to produce drought-resistant soybean. We first have to figure out what genes are responsible for drought stress tolerance. One way of doing this would be to expose plants to a drought-stressful environment and also normal conditions. When the plants are under stress, certain genes will be activated and upregulated. Expression of certain genes are altered during adaptation to drought stress situations. We can then pick these genes to compare their expressions during conditions of stress versus normal conditions. Another way to figure out exactly what genes are responsible for what function is the use of molecular linkage maps to identify quantitative trait loci. Quantitative trait loci, also called QTLs, are a specific region on chromosomes which have the genes controlling the traits. Using QTL mapping, we can identify the parts of DNA that are associated to genes underlying a specific trait. In the case of soybeans, we would use molecular linkage maps to identify the quantitative trait loci associated with drought resistance. Using molecular linkage maps to identify the regions of DNA associated with drought stress and drought resistance, we can then use genome editing technology to alter the arrangement of nucleotides within DNA to produce a soybean crop that can resist drought. This idea holds true with any crop and any trait. Whether disease or environmental factors, we can use genome editing technologies to create crops that can withstand any conditions. So, as seen here, food insecurity is a big issue. There are so many people in the world that are starving. And while there are many reasons for this, there is something we can do to at least help a little. We can produce crops that can resist anything using genome editing technology. This field is still being researched and there is so much unknown, but we have to start somewhere. The use of genome editing technology is the start of something great. If we can produce crops that can resist disease or drought or too much rain or pH conditions, then we can produce more crops. We can produce more food. More people can be fed, and food security can be improved. Here's a list of all my references I used throughout my presentation. Thank you for listening.